Understanding the difference between a gene and a chromosome. 1. Definition and function. Genes and chromosomes are fundamental components of genetics and play crucial roles in determining an organism's traits and genetic makeup. A gene is a segment of DNA that contains the instructions for creating specific proteins or RNA molecules. Genes encode traits such as eye color, blood type, and susceptibility to certain diseases. Chromosomes, on the other hand, are thread-like structures made of DNA and proteins that carry genes. They exist in the nucleus of cells and are responsible for organizing and packaging genetic material. 2. Structure and composition. Genes are localized segments of DNA that consist of specific sequences of nucleotide bases, adenine, A, thymine, T, cytosine, C, and guanine, G. These sequences determine the genetic code and instructions for protein synthesis. Chromosomes are larger structures that consist of condensed DNA wrapped around proteins called histones. Humans have 23 pairs of chromosomes, with one set inherited from each parent, totaling 46 chromosomes in each cell, except for gametes, which have 23 chromosomes. 3. Genetic inheritance. Genes are inherited from parents and passed down through generations. Each parent contributes one copy of each gene, which may be dominant or recessive, influencing the traits expressed in offspring. Chromosomes carry multiple genes arranged linearly along their length. The inheritance of chromosomes follows Mendelian principles, where chromosomes segregate independently during meiosis to produce genetically diverse offspring. 4. Organization and packaging. Genes are organized along chromosomes in a linear fashion, with each gene occupying a specific locus, position, on a chromosome. Chromosomes are organized into pairs, with homologous chromosomes, one from each parent, carrying corresponding genes. The packaging of DNA into chromosomes allows for efficient storage and transmission of genetic information during cell division and reproduction. 5. Role in cell division. Genes and chromosomes play crucial roles during cell division processes such as mitosis and meiosis. During mitosis, chromosomes replicate and condense into visible structures that ensure each daughter cell receives an identical set of genetic material. Meiosis involves the formation of gametes, sperm and egg cells, where chromosomes undergo recombination and segregation to create genetic variation. 6. Variation and mutation. Genes are subject to mutations, which are changes in DNA sequence that can alter protein function or gene expression. Mutations can occur spontaneously or due to environmental factors and may lead to genetic disorders or adaptations. Chromosomal abnormalities, such as deletions, duplications, or translocations, can also occur, affecting gene dosage and causing genetic disorders like Down syndrome or Turner syndrome. 7. Study and research. Genes are studied through molecular genetics techniques such as DNA sequencing, PCR, polymerase chain reaction, and gene editing tools like CRISPR-Cas9. Researchers investigate gene function, regulation, and interactions to understand biological processes and diseases. Chromosomes are studied through cytogenetics, which analyzes their structure, number, and abnormalities using techniques like karyotyping and fluorescent in situ hybridization, FISH. 8. Evolutionary perspective. Genes and chromosomes play pivotal roles in evolutionary biology by contributing to genetic diversity within species and adaptation to environmental changes over time. Understanding the evolution of genes and chromosomes provides insights into speciation, phylogenetic relationships, and the genetic basis of evolutionary traits.